Hello everyone. So today we are going to discuss fluorescence microscopy. Now before the discussion of fluorescence microscopy, let us first look at the principle of fluorescence. What happens in fluorescence? So first you can notice that there are some energy levels. This is the ground state energy level in an atom and these two are the excited energy levels, right? So, and these two are the electrons. Now, these two electrons are in the ground state energy level. Now, when you excite these two electrons with an excitation light, what happens? These two electrons absorb that energy of the excitation light and move into a state of high energy. So, what you are doing is that you are putting an excitation light which is of high energy. That means of shorter wavelength. Why? Because we know the formula E is equal to hc by lambda. We know this formula, right? So E is equal to hc by lambda means E is inversely proportional to the wavelength lambda. So if E increases, that is the energy of the light increases, then wavelength of the light decreases and vice versa. So you are putting an excitation light which is of high energy, that means the light is of shorter wavelength, and the electrons in the ground state are absorbing that energy of the excitation light and going from a state of lower energy to a state of high energy. It remains, the electrons remain in the excited state for some time and thereafter it tends to return back to the ground state energy level. So what does it do? After some time it loses the absorbed energy, right, and comes back to the ground state. So while the electron loses its absorbed energy, it emits light, and this emitted light is what we call as fluorescence. Now this emitted light is of lower energy and so of higher wavelength. Because I have already told you E is related to hc by lambda. So if E is lower, lambda will be higher. So this emitted light is actually the fluorescent light, and so the fluorescent light is always of higher wavelength than the absorbed light, but the fluorescent light is always at a lower energy level than the absorbed light. So this is the principle of fluorescence. If you look at in this figure also, here the electron was in the ground state. Now you excite this electron with an excitation light which is of higher energy and shorter wavelength. So what happens? Electron is moving from this energy level, ground state energy level, to an excited energy level. Here in the middle figure, you will notice electron has moved from a lower energy level to a high energy level after absorption of the energy of the excitation light. Now after some time, when it returns back to the lower energy level, what will happen? It will emit the absorbed energy in the form of light, which is called as fluorescent light. This light is of lower energy and hence higher wavelength. This is the principle of fluorescence, and on this principle the fluorescence microscopy works. So what happens? We take a mercury arc lamp, which is the light source here. In case of fluorescence microscopy, we take a light source, preferably a mercury arc lamp which emits the light. Okay, and here we have an exciter filter or excitation filter. The function of this filter is to remove the longer wavelength. So it filters longer wavelengths and only permits the passage of shorter wavelengths. So by using this filter, you can remove all the longer wavelengths, all the undesired long wavelengths of light, and only your desired short wavelengths of light will pass. So here you will notice that light is passing from this excitation filter. Here it is going and then it is reflected by the dichroic mirror. Now the function of the dichroic mirror is that for short wavelengths, it reflects shorter wavelengths, but for longer wavelengths, it transmits the longer wavelengths. So it has two functions. First, if you have given a shorter wavelength, then it will reflect those shorter wavelengths of light. Okay, but if you have given longer wavelengths, then it will transmit the longer wavelengths of light. So depending upon the wavelength of light, this dichroic mirror has two different functions. So the shorter wavelength, on reaching the dichroic mirror, according to this property, are reflected and then they are falling onto the sample. Okay, now this shorter wavelength of light is functioning as the excitation light, which we discussed previously. So the sample absorbs this excitation light, and in the sample the electrons absorb the excitation light and move from a ground state to an excited state. After some time, when the electrons in the sample move back from the excited state to the ground state, they release the absorbed energy in the form of fluorescent light. So if this is the fluorescent light, I will choose a different color. If this is the released fluorescent light, then what happens, the sample will emit this fluorescent light and it will directly form on this dichroic mirror, dichromatic mirror. Because dichromatic mirror transmits the longer wavelengths, and fluorescent light we already know is of longer wavelength. So what will happen, it will directly pass the longer fluorescent light, longer wavelength fluorescent light, and it will directly fall on the eyepiece. Okay, so this is the main principle of fluorescence microscopy. So what you are doing, you are selecting shorter wavelengths of light by using an excitation filter. You are selecting shorter wavelengths of light and you are allowing that short wavelength of light to fall on this sample. Now this sample has number of electrons which are absorbing the shorter wavelength of light and moving from a ground state to an excited state. After some time, when the electrons are releasing their absorbed energy, they emit the absorbed energy in the form of fluorescent light. We already know that fluorescent light is always of longer wavelength. So what will happen, on releasing the fluorescent light, this fluorescent light directly form on the dichromatic mirror. Now because the dichromatic mirror transmits longer wavelength, it will straight away pass the longer wavelength fluorescent light and it will directly form on the eyepiece. The image that you observed is here. Here you put your eye and here you observe the image. So this is the main principle on which the fluorescence works. Particularly the shorter wavelength of light that is particularly chosen for fluorescence microscopy most of the time is preferably it is blue light. This is the shorter wavelength of light that is most of the time chosen, and the longer wavelength of light that passes through the dichromatic mirror, preferably it is green light. Okay, this is the main principle on which fluorescence microscopy works. So the most common type of fluorescence microscopy is called as epifluorescence microscopy. Here I have given a list of some of the most common fluorochromes. What are fluorochromes? Fluorochromes are those compounds which absorb light. Okay, so and which emit fluorescent light. So fluorochromes are those compounds which absorb a certain amount of excitation light and in turn emit some fluorescent light. So these are called as fluorochromes. So number one, we have acridine orange. Now acridine orange is particularly used for staining DNA and it gives orange color. Then we have diamidino 2-phenylindole or DAP1 or DAPI. So this particularly stains DNA and it fluoresces green. So these two are very important. In the lab we use either of the two, okay, for staining DNA. Either we can use acridine orange for staining DNA, which then gives orange light, or we can use DAP1 or DAPI for staining DNA, which gives green light. DAPI is actually diamidino 2-phenylindole. Then we have another fluorochrome which is called as fluorescent isothiocyanate, FITC. It is mostly used for immunological assays. it is attached to antibodies or DNA probes. So it is mostly used for immunological assays and it gives green fluorescence. Then last one we have tetramethyl rhodamine isothiocyanate or rhodamine. So it is also used for the antibodies. It is attached to antibodies. It is mostly used for immunological assays and sometimes it is used for staining actin filaments as well, and it gives red color. So these last two are mostly used for immunological purposes. Okay, mostly used for immunological purposes, and the first two are mostly used for staining the DNA. So in the below pictures I have shown some of the microscopic images of how fluorescent microscopy looks like. So here you will notice that these are the fluorochromes or the compounds which give fluorescent are stained as green. These are actually live cells. Okay, so these are actually live cells which are giving green color under fluorescence microscopy, and the ones which are giving red color like this one, so these are actually dead cells. Because they are dead cells, so they couldn't absorb the excitation light and go into a state of higher energy and then emit the fluorescent light. So that is why they are not giving sort of the color that we expect. So the green cells that you can see here are the live cells, and the dead cells are the ones that are and the red cells are the ones that are dead cells. In the second figure you will notice that these are actually mycobacterium species, and this is actually acid fast staining microscopic image, and the dye used is called as auramine dye. And the last one is an immunological assay. So this is actually the fluorescence antibodies tag specific molecules. So what happens here is you have the antibody here and you have the fluorochrome. So this is the fluorochrome attached to the one end of the antibody, and this antibody binds to some antigen. This is the antibody binding to some antigen, say a bacteria, it can be a bacteria, and on proper binding of the antibody with the antigen, what happens? This antibody releases light. Okay, the fluorochrome releases light, and that you can observe under fluorescence microscopy. So this is also used in immunological assays. Fluorescence microscopy is also used. So this is a figure showing the immunological assay. This third picture.